All right, welcome. Today, we are diving deep into something that's happening inside you billions of times a second, protein synthesis. It's this incredible journey of how the instruction manual written in your DNA gets turned into the actual stuff that makes you, well, you.
So just think about that for a second. How does a simple chemical code, just a long string of letters tucked away inside every single one of your cells, become all the different parts of you? Your muscles, your hair, the enzymes in your gut? That's the big question we're going to unpack today.
So here's our game plan. We're going to follow the whole process from start to finish. First, we'll talk about why proteins are such a huge deal. Then we'll go to the source code itself, the DNA master blueprint. After that, we'll see how a copy of those instructions gets made, watch it get built at a molecular factory called the ribosome, and finally see how a simple chain becomes a finished product. It's a whole journey from blueprint to building.
Okay, first things first. We have to establish just how critical proteins are. You can think of them as both the construction workers and the building materials for the entire city that is your body. Without them, absolutely nothing gets done.
I mean, just look at the variety of jobs they do. It's kind of mind-blowing. Proteins are the enzymes that kickstart all the chemical reactions that keep you alive. They form the very structure of you. We're talking about stuff like the collagen in your skin and the keratin in your hair. They act as messengers, like the hormone insulin, carrying signals all over the place. The bottom line is, for any kind of growth, repair, or just basic day-to-day function, you absolutely need proteins.
So if proteins are the workers, where do they get their orders from? That brings us to the DNA, the master blueprint. This is the complete, original set of instructions, and it's kept safely locked away in the cell's nucleus, because you do not want this thing getting damaged.
And the language this blueprint is written in is called the genetic code. It's wild. It's written in sequences of chemical bases, and the cell reads them in little three-letter words. We call these words codons. Each codon is a specific command that says, add this one particular amino acid, which is, of course, the basic building block of a protein. It's pretty much a universal language for almost all life on Earth.
Okay, so here's our first big problem. The master blueprint, the DNA, is way too precious to leave the safety of the nucleus. So to build a protein out on the factory floor of the cell, we've got to make a cheap, disposable copy of the instructions. This first stage is called transcription.
And this entire copying process, it has to happen right where the blueprint is stored, inside the cell's main office, the nucleus. Think of it like a high-security vault where the most important information is kept under lock and key.
So let's walk through this. Picture this. First, the DNA double helix unwinds and unzips, but only at the specific gene we need to copy. Then, an enzyme called RNA polymerase, our little molecular photocopier, comes in and attaches to the DNA. It zips along the strand, reading the code and building a copy out of complementary bases. This new copy is called messenger RNA, or mRNA for short. Once the copy is made, it just peels away, and the original DNA zips itself right back up, totally unharmed and perfectly preserved.
But wait, this fresh photocopy isn't quite ready for the factory floor yet. It needs a little bit of editing first. Any useless, non-coding sections, called introns, get snipped out. This is a process called splicing. Then, a protective cap gets added to one end, and a long, stabilizing tail gets added to the other. Now, finally, we have a mature, ready-to-go mRNA molecule. It's ready to leave the main office.
All right, with our polished instructions in hand, it is time to leave the nucleus and head out to the factory floor to actually build something. This second major stage is called translation. It's called that because we are literally translating the language of the genetic code into the language of proteins.
And the factory floor for this whole operation is a tiny little structure called the ribosome. These are these amazing molecular machines that are just floating around in the cell's cytoplasm. This is where the real construction work happens.
So let's meet the construction crew. First, we have our mRNA. That's the photocopy with all the instructions. Then we have transfer RNA, or tRNA. Think of these like a fleet of little delivery trucks. Each one is designed to carry one and only one specific amino acid. And finally, we have ribosomal RNA, or rRNA. This stuff actually makes up the factory itself. It's what the ribosome is built from.
So here's how this incredible assembly line works. The mRNA blueprint slides into the ribosome. The ribosome reads the very first codon, which is almost always AUG, that's the universal start signal. Instantly, a tRNA delivery truck with the matching code arrives, carrying the first amino acid. The ribosome then forges a special bond, a peptide bond, linking it to the next one, and that starts our protein chain. Then it just chugs along to the next codon, and the whole thing repeats. Read, deliver, link.
read, deliver, link. Over and over, building the chain one amino acid at a time until, bam, it hits a stop codon. That's the signal. The factory shuts down and the finished chain is released.
But hold on, we're not quite done. What we have right now is just a long, floppy chain of amino acids. It's not a protein yet. To actually do its job, it needs some serious finishing touches. This is what really brings the protein to life.
And here's what is maybe the most crucial step of all. That chain has to fold itself into a very specific, incredibly complex 3D shape. And everything, I mean everything, depends on that shape. The shape determines the protein's function. Sometimes it gets sent off to other parts of the cell, like the Golgi apparatus, for some final modifications and packaging. And it's only after all of this intricate folding and processing that it becomes a fully functional protein, ready to get to work.
And there you have it, the entire spectacular process. You go from a simple 4-letter code safely stored in your DNA all the way to a complex, 3-dimensional, functional machine that carries out some vital job in your body. It is an absolutely beautiful and elegant system.
So let's just boil it all down one last time. Super simple. The two main stages are transcription, which happens in the nucleus to make the copy, and translation, which happens at the ribosome to build the actual thing. That's the whole journey, from blueprint to building.
It's an unbelievably precise and elegant process. But it does leave us with one last really fascinating question to think about. What happens if there's a typo, a mistake in that original blueprint? What happens if just one of those millions of letters in the DNA code is wrong? Well, that's a story for another time.